Good afternoon, everyone. Thank you very much for joining today's session. This pitching session will be focused around strengthening the European ecosystem for advanced therapy medicinal products, ATMPs, and other innovative therapeutic modalities for rare diseases. We'll have 10 presentations today, and I'd like to remind you, if you want to interact with the other participants, you can use the chat on the right top corner. Before starting the session, I'd like to remind you that this is being recorded, and both the recording of the session as well as the presentations will be shared later on the IHI's website, as well as the B2Match platform. And how you can contact the other presenters. So you can do it via the pictures link on the top navigation, or via the agenda. You also have the list of pictures for this session. So you can contact them directly via the B2Match platform. So without further ado, I would like to ask Els to start with her first presentation. Thank you very much, everyone.
Good afternoon, everyone. I'm Els Henkaerts from KU Leuven in Belgium, where we're in the process of setting up a gene therapy center of excellence. And to support this endeavor, we're looking to join an IHI consortium. Next slide, please. So we've recently been awarded a 9.1 million euro investment by the European Commission in the context of the Recovery and Resilience Facility to build an AAV gene therapy facility. And this entails a state-of-the-art facility enabling preclinical development, manufacturing, process development, and AAV manufacturing innovation. To make the most out of it, we would like to fully align this with industrial practice so that we can support end-to-end gene therapy development. That's, of course, a very challenging endeavor, and we think that the solution would be is to get access to a network of biotech and pharma partners with relevant expertise. And the impact from being able to join this network is cost-effective integrated gene therapy development for rare diseases. In parallel, we're also setting up a scientific center of excellence for AAV discovery research. And this is basically to start tackling those challenges that we're currently facing in the field of AAV gene therapy. Amongst those are off-target toxicity, longevity of transgene expression, and manufacturing yield that it's not entirely optimal to really support manufacturing for larger patient populations. One of the challenges, or several of the challenges that we're facing here, is early access to regulatory advice, CMC advice, and complex data analysis. And here also the solution would be that we get access to a network of biotech and pharma partners with the relevant expertise. And again, the impact would be, safer, cost-effective, and long-lasting AAV gene therapy products. Next slide, please. So just to give you a flavor of the activities that we're doing at the moment, so we do AAV production, research grade. We do that both in adherent cells as well as in suspension. And then by the end of the summer, with the help of our facilities that we've been able to build, we will be able to go up to 50 liters. We also do process development, so improving the base process, as well as early CMC that's associated with gene therapy asset development. And then side by side, we also do manufacturing innovation, where we are improving the plasmids that are used for vector production, using information from our insights into AAV biology, but we're also exploring alternative DNA technologies to make the process more lean and more effective. We're also considering additives as well as cell line development, again, all to improve the yield of AAV manufacturing. And we also have the ability to do preclinical studies starting from a solid proof of concept. In the Scientific Center of Excellence, the activities to support the off-target delivery range from protein engineering, early CMC, in vitro and vivo modeling, proof of concept and disease models. For longevity of expression, we're focusing on viral epigenetics, where we again have a range of activities ranging from vector design, in vitro and vivo modeling, high-throughput sequencing, and bioinformatics. AAV manufacturing entails activities from molecular biology, multiomics, mathematical model, and synthetic biology. Next slide, please. And so the expertise and resources offered by us is, long-standing expertise in AV biology, molecular biology in general, and vector design, process development, research-grade manufacturing, preclinical development, and protein engineering. So as I already mentioned, we have a new facility that's over 1,000 square meters that supports, many of the activities that I mentioned. Plus, we also have access to state-of-the-art core facilities, including multi-omics facilities, as well as high-end biophysics and microscopy facilities from KU Leuven. We have a team of 25 people, skilled scientists, collectively experience over 30 years of AAV research. We are supported by one of the best tech transfer offices in Europe. We also have business development expertise within the team. And we are already currently working with several partners in the biotech and pharma, and we have built a network of clinicians and other academic partners to really support AAV gene therapy development. Next slide, please. The next slide, please. Thank you. So here we can just basically summarize that we're looking for expertise from pharma partners with experience in all aspects of AAV gene therapy development, biotech companies providing complementary technologies, and academic partners providing complementary expertise and technologies. And with this, I'd like to end my pitch.
Great. Thank you very much, Els, for your presentation. I would like to call the next speaker to the stage, Claudia.
Yes, I'm here. So good afternoon to you all. Welcome to our pitch entitled Bio-Inspired Cell Expansion Platforms to Promote the Feasibility of Autologous Transplantation for Rare Diseases, namely bone marrow failure syndromes. Next slide, please. So allogeneic bone marrow transplantation is considered a curative treatment for many conditions, although it is unfeasible for patients who lack a suitable donor, which compromise the treatment of these conditions. On the other hand, autologous transplantation has been challenging due to the reduced numbers of hematopoietic stem and progenitor cells in the bone marrow of these patients. So our proposed solution is to contribute towards a better understanding on the molecular and cellular differences in the defective bone marrow and microenvironments in the context of these diseases, towards the establishment of a bio-inspired cell expansion device, which could be able to recreate the elements of a functional niche in order to expand autologous stem and progenitor cells. We believe that the success of our project could largely improve the feasibility of the application of autologous transplantation as a first-line therapy for patients in which allogeneic transplantation is not viable. In a more precision medicine perspective, we believe that this expansion device can be used to cultivate cells from the patient in order to identify new orphan drugs or optimize therapeutic regimens of currently administered pharmaceuticals. We also believe that this device can have a high commercialization potential, especially if we consider other potential applications, namely in which we have to surpass suboptimal cell doses. Next slide. So here you can see a graphical abstract of the proposed activities in this project. So we expect to have access to bone marrow samples of both patients and also healthy controls. In a second level, we want to characterize the dysfunctional bone marrow niche at the cellular and molecular level using different techniques. And then in a third level, we want to develop a perfusion chamber in which hematopoietic stem and progenitor cells could be expanded in order to reach clinically meaningful cell numbers for autologous transplantation. Next slide, please. So who are we? IST has been recognized to be an international leader in research and training programs in the field of biomanufacturing. Over the years, we have gathered expertise in isolation, characterization of human hematopoietic stem and progenitor cells, and also mesenchymal stromal cells of human origin. Also on the GMP-compliant cultivation of these cell types using genogenic-free culture conditions, 2D and 3D co-culture settings, also the development of bioreactor platforms for stem cell cultivation, and more recently, the development of biomimetic extracellular matrix-based scaffolds. Of notice, we have a clinical partner, the Pediatric Hematology Unit, who is the national coordinator for the European Working Group of Myelodysplastic Syndrome and Severe Plastic Anemia in children and adolescents. Next slide, please. So who do we want to collaborate with? We want and we are seeking for fundamental research centers dedicated to hematopoiesis and hematopoiesis stem cell biology, who can join us. Also, we are seeking for centers with a large expertise in multiomics and bioinformatics, clinical research centers devoted to the study of hematological rare diseases, advanced cell therapies SMEs, scientific and regulatory affairs consultant, and also patient associations. Thank you for your attention. If you are interested, please contact us.
Great. Thank you very much, Claudia, for your presentation. I would like to ask then Volker.
Hello and welcome to the presentation. Next slide, please. Can you hear me? So what we would like to propose to a consortium would be to use molecular imaging to study an ATMP. And this ATMP could be gene therapy, it could be oncolytic viruses, it could be cell therapies. But for all of these therapies, we definitely need to better understand on a molecular level what is happening in test animals and also in the patient. We need to closely understand how they are trafficking, how they are infiltrating, how they expand, or if they die. So this can be best studied using molecular imaging. Next slide, please. And what we would like to propose in this regard is a novel reporter gene that we created. And we are very happy with the results and believe that this novel reporter gene is best in class regarding sensitivity and specificity. And now I will come up with a few examples of what we can image. Next slide, please. So the first example matches the first presentation. We just had a look on basic AAV9 gene therapy. So we produced AAV9 vectors that encoded the reporter gene and then infused systematically into mice via the tail vein. And in the middle, you see a presentation or a picture that is from a publication using bioluminescence imaging. And we only have rough expression levels, but it's not depth independent. On the other hand, on the right, we see some results that we can obtain from AAV9 transduction using our reporter gene. We nicely see transduction of the heart, brown fat tissue, some muscles, some neurons, adrenals, and the liver. Next slide, please. We can also look at dose response. So if we are injecting different titers to these mice, we see that the more virus we are giving or viral vector we are giving, the more transduction we are obtaining. And this also tends to be nicely linear, as we see on the upper panels. Also, this correlates nicely when we go for immunohistochemistry on a molecular level. So transduction levels can really be measured using this PET technology. And PET is really of advantage here because it's inherently quantitative. So the unit that we are measuring in these scanners is the case per volume. Next slide, please. Another application scenario, which would be in this case cell therapies, is CAR T cell therapy. And here we used a Raji model, which is a lymphoma going to the bones. And these mice were then treated using CD19 CAR T cells. And on the lower panel, we see time course imaging over 30 days. And we see at day 4, we see infiltration in the spleen. And then from day 8 on, we see the CAR T cells producing some signals in the bones, in the hollow bones. And this is then also increasing over time. We can have a general understanding of how CAR T cells are expanding in individual animals. And this can be used to understand better therapy. Next slide, please. On the other hand, what we see, and here we see a PET MR of the spine of these animals. We see these small dots, and when we go then and look at the spine in immunohistochemistry, we see individual infiltration in these vertebrae of the animal. Once click, please. And then if we correlate this with the PET, we see that the correlation with the PET is quite nice. And so these small dots that we see are individual infiltration into vertebra. Next slide, please. So one important point for the development of ATMPs is definitely 3 Rs in animal protection. So this is required by law, and we believe that molecular imaging is one of the best ways to achieve reduction and refinement. And that's also the reason why we are working on a project to develop this technology with the NC3Rs. And we can do mice, rat, and pig imaging. Next slide, please. So we would like to offer this technology to a consortium, and you have new use cases. Next slide. Yeah, this is the people that contributed, and if you're interested, we would be happy to have a discussion. Thank you a lot.
Thank you very much for your presentation, Volker. I would like to ask the next speaker, Tero, to join us.
Okay. Thank you very much for the opportunity to present today. What I'm offering is a single asset, a single molecule for rare disease called epidermolysis bullosa. So our molecule is a recombinant protein, and we have demonstrated in the proof of concept studies that we can improve the survival of the animals with the lethal form of the epidermolysis bullosa more than with any previously tested therapeutic, and we want to advance it to epidermolysis bullosa patients. Next slide, please. So the molecule is a multifunctional recombinant protein, and our sole focus is to introduce it as a drug for rare skin disease called epidermolysis bullosa. Epidermolysis bullosa is a lethal, genetic rare disease, and it's an unmet medical need at the moment that there are no drugs available to treat epidermolysis bullosa, and we hope to introduce a potentially life-saving drug that improves the prognosis of the patients with the epidermolysis bullosa. Next slide, please. We have carried out the proof of concept studies. Now we need to move our recombinant protein production into the GMP facility. So we need to create the master cell bank, do the actual protein, recombinant protein expression and purification. Then we need to do pre-IND drug development, PK and tox studies, and put the data packets all together for IND approval, and then carry out phase 1 and phase 2 clinical trials in epidermolysis bullosa patients. Next slide, please. So this is what we offer for the drug development. We have identified the final drug product. We have demonstrated the efficacy of the molecule in the most representative disease models, and we have obtained the best-in-class position. So proof of concept studies have already been performed. We have the orphan drug designation awarded, and we have worked with the CDMO company to confirm that we should get commercially viable high expression yields of our recombinant protein from the mammalian expression system. We have also recruited the leading academic institute from Europe, from Paris, to carry out phase 1, phase 2 clinical trials, and we are currently writing the protocol for these studies. Next slide, please. So this is what we are looking for. We definitely need funding. We are looking for a pharmaceutical company as a funding and research partner. At the same time, we want to carry academic research to expand the indications of our molecule beyond the rare disease. So we are also interested to collaborate with the research institute to do academic research. We need more expertise on drug development to join our drug development team. And as it's rare disease where we are focusing, we want to engage with the patient advocate groups and organizations. Next slide, please. Thank you very much for your attention.
Thank you, Tero, for your presentation. I would like now to call Raymond to the stage, please.
Thank you very much. Indeed, so my name is Raymond Schifflers. I work at the University Medical Center in Utrecht, and especially in my lab, we work on lipid-based nanomedicines. Next slide, please. And what we're trying to solve, or the challenge that we're trying to solve, is that nucleic acid therapeutics are attractive ATMPs, but at the same time, they have difficulties in reaching their intracellular site of activity. And what my lab does is to design, make, and evaluate lipid-based nanomedicines that functionally deliver these nucleic acid therapeutics intracellularly, and that can be both actively targeted or passively targeted. They can be both natural, and those are exosomes or extracellular vesicle lipid-based nanoparticles, or synthetic, and those are the lipid nanoparticles that are currently, for example, in the COVID vaccines. And of course, you can also envision hybrids of the two. Next slide, please. So our main activities are the design, synthesis, and characterization of these lipid-based nanomedicines, and evaluate their performance in sensitive in vitro and in vivo reporter systems that we've made, so CRISPR-Cas-based or CRELOX-based. And we also test in vitro and in vivo therapeutic testing, and there's a wide network on the campus for that. For example, in the field of metabolic diseases, where rare diseases like MMA and LCHAT, there are patient organoids that are being developed, and there are zebrafish and mouse models of those. We have an active program in cardiac rare diseases, for example, the phospholamban mutation, which is very prominent in the Netherlands. There are also patient organoids and mouse models developed, and also for rare tumors, for example. Next slide, please. So as an example of the type of work that we do, in panel A, you see a lipid nanoparticle composition, the sort of sampling of the design space. And then in panel C, we tested 33 of those formulations to look at the immune response that we can generate with different compositions of lipid nanoparticles. And I think, well, it's clear that the lipid nanoparticle composition dictates the immunological response. And that you can also model, and then you arrive at the model presented in D, where you see the green areas. Those are the sweet spot compositions that really result in a very high immune response. And in this case, we could also relate that to splenic expression, actually, of the messenger RNA that we tested. Next slide, please. We also performed a head-to-head comparison in very sensitive assays, so where we looked at the lipid nanoparticles, those are the current state of the art, and compared it to the extracellular vesicles, exosomes, that created a lot of excitement. So on the right, you see the CRISPR-Cas-based reporter assay that we developed, where the successful delivery of a single guide RNA results in a color change in the cells. And then we either titrated the lipid nanoparticle state of the art to the levels of RNA that we find in extracellular vesicles, or we spike them in. Doesn't matter, the end result is the same. And we find, surprisingly, that the LNPs are about 1,000-fold less potent compared to the extracellular vesicles. Those are already active in the gray area, whereas the green doses in panel E and F are the ones that result in activation of the reporter system for the synthetic system. So that offers a lot of inspiration to explore how this phenomenon occurs later on. Next slide, please. So the expertise that we have a local network, as I just explained, but I'm also a coordinator of the expert consortium and the Be Smart consortium. Those are consortia where we design and develop nanoparticles for nucleic acid delivery. We also have an active program on quality control and manufacture, and ultimately leading to the preparations for clinical trials. So we have the whole chain, actually, of development of these particles. And within my lab, then, we have a fully equipped lab for chemistry, nanomedicine characterization, and the vitro vivo testing, and of course the clinical partners on site. What could also be of interest, actually, is the fact that we've recently been awarded a national grant. That's the Nanospresso NL. And there we actually, within the framework of rare diseases, I think very relevant, trying to use bedside preparation of lipid nanoparticles to actually make a single dose for a single patient at the bedside, without the scaling up and manufacture challenges that are normally associated with rare therapeutic lipid nanoparticle development. Final slide is the next slide. The expertise requested. I think we have a strong network within the consortia that I just sketched of innovative SMEs and research institutes and universities. So I think the biggest expertise that is required is that of large companies that have the experience to guide a product, well, basically from the design all the way to clinical trials. And of course, that would also be the in-kind matching that is required for IHI calls, and that is something that we currently have not been able to achieve. So we offer a very strong collaborative environment all over Europe in existing consortia, and what we're looking for is large companies with interesting therapeutic payloads. Thank you very much.
That's great. Thank you very much for your presentation, Raymond. I'd like to ask then Felix to join us on our virtual stage.
Yes, hi. This is the bioMérieux presentation. I am Felix Montero, Scientific Director for the Pharmaceutical Quality Control business. Next slide, please. So the challenge we are seeing today in the ATMP field is the lack of availability of relevant analytical tools that are essential for the manufacturing and process development controls in order to have products of good quality and safety and enable all the process improvements. So we have identified some main challenge in the whole ATMP ecosystem regarding the evolution of the regulatory landscape, the rapid advance in manufacturing technologies and products that have a very complex mechanism of actions, and this is really inherent to the raw materials that we are using. And this is just a few examples. So, for example, the lengthy and complex manufacturing process, and in some cases, for some of the products are developed, it's extremely short shelf life of these products to a few days. The low production volumes bring in some complexity in terms of sensitivity of the analytical assays, and also complex raw materials. So in addition, whatever is an autologous or an allogenic approach to manufacture these ATMP products, it requires a high traceability, data integrity, and management of the scalability of the products depending on patients' demands. Next slide, please. In that order, so bioMérieux objectives and expected outcome is to develop and improve technologies and process analytical tools, methods, and assays as are needed for the development and the production of these ATMPs. And these analytical methods are fully adapted to the ATMPs, and these methods must compensate all the challenge we described before. So the contribution of bioMérieux to the outcome of the program shall encompass the adding of rapid and easy-to-use analytical technologies in order to bring improved efficiency to the manufacturing process and developments, simplified workflows, and ultimately improve patient safety and patient access to these technologies, contributing also to making a sustainable and accessible network of interconnected center of excellence in the ATMP, and also contributing to eligible regulatory landscape. And the work that can be provided could be also establish some of the reference standards in the future. Next slide, please. So the main activities as IHI partner from bioMérieux is really to identify the unmet needs in terms of analytical methods development, to select and test breakthrough technologies allowing the rapid implementation in ATMP cell-based processes, also to contribute to the scouting and networking of Center of Excellence, and contribute to developing standards and fits into the European regulatory landscape. Next slide, please. So the expertise that we are offering is really experience in the development of analytical methods designs. We are also leaders in the medtech industry and industrial applications, and this is fundamentally supported by 4 different pillars related to our experience in the development of immunoassay, molecular biology, microbiology, but also cellular biology. Next slide, please. So the expertise and resource bioMérieux is offering here is really to improve technologies, process, analytical tools, methods, and assay. As I explained before, is our expertise in medtech development and diagnostics, medical and pharmaceutical expertise, expertise in immunomonitoring approaches, technologies for potency monitoring, and also working with different regulatory aspects of the analytical methods. The experience we have working with ICH Group, Pharmacopoeias, and others, but also our experience interacting in an ecosystem composed by different stakeholders. And this is only one example of our collaborations that we have currently with NIBR, Catapult, or NextGen. Resource offer an expertise in project management, project communication, but also contribute to the knowledge in terms of legal expertise, ethical and regulatory questions. Next slide, please. And the expertise requested here is really bioMérieux is interested in joining existing consortium as a partner. As such, we will be looking for the following profiles within the consortium. So ATMP developers, pharma industry, biotechs and medtechs, CROs, CDMOs, patient organizations, but also academic center of excellence in cell and gene therapy. Next slide. Yep, that's it. Thank you very much for your attention.
Thank you, Felix, for your presentation. I'd like to call then Michael and Dirk to stage.
Yes, hello. So we like to answer call 3 by the proposal of a European research and development ecosystem for rare diseases. And I have to say that in this effort, Pfizer and UCB are assembling a consortium to address this proposal. I'm happy to say that INSERM also has agreed to coordinate this activity. Next slide, please. So the vision behind there is really to establish strategically targeted networked centers of excellence that are focused on ATMP technologies, ultimately benefiting patients through the increased availability of effective and safe treatments for rare diseases. Overall, the idea behind this is that Europe will or can become a more attractive and competitive space for developing ATMPs for rare diseases and beyond. The expected outcome includes sustainable networks of excellence, agreed standards, and as we all know, these ATMPs very often suffer from the absence of standardization, a streamlined and more transparent regulatory pathway, improved technologies and processes, analytical tools, methods, and assay. So really what we hope is that development will be streamlined thanks to enhanced scientific and technological processes, and the ability of networks of excellence to improve the development of specialized rare disease treatments is anticipated to benefit a broader range of diseases beyond rare diseases once these networks are established. Again, all of this together, we hope that with this proposal and in the implementation, Europe can become an attractive area for the development of ATMPs for rare diseases and beyond. And here I would like to hand over to Dirk, who will tell you some more specifics.
Thank you, Michael. Next slide, please. So as Michael explained, the starting point of the consortium was really the observation that there is this emerging science, ATMP, cells and gene-based therapies, but there are still important scientific, technological, and also regulatory hurdles that are still perceived to be limiting factors for the rapid and cost-effective development of these assets, of these technologies. And we believe by building these networks of excellence around some of these technologies that we can hopefully translate and scale and start validate some of these technologies so that they can be used for the development not only for one single asset or one single rare disease, but hopefully with the potential to address multiple types of rare diseases. Next slide, please. Our current thinking, and based on discussions we already had within with industry consortia, is that we would like to focus the networks of excellence around in vivo technologies. Our current thinking is centered around messenger RNA technologies or platforms to really allow for enabled and targeted delivery. We'd like to establish another network of excellence around nanoparticle development, especially for lipid nanoparticle delivery of RNA type of platforms, and also another network of excellence around in vivo gene therapy, the AAV platform, but also gene editing type of platforms, such as gene or base editing. What will be critical for the success of these consortia is to make sure that we have a strong regulatory alignment, not only with European regulators, but also with FDA and maybe other regulatory instances, that we have a close connection and the work being done around these proposed platforms, that the work starts to be agreed, validated according to standards being discussed and validated with regulatory authorities. And we believe that if with this consortium, we have an opportunity to really streamline some of the development, streamline some of the regulatory expectations, so that again, if we can start validating some of these technologies, that they can be used not only for one single disease, one single asset, but hopefully for multiple types of rare diseases. And as Michael mentioned, this ultimately will not only be beneficial for diseases in the rare disease space, but hopefully will have the potential to also unlock some other unmet medical needs in other disease areas. Next slide, please. What we have done so far, we already secured a very strong interest and also contributions from multiple industry partners, FPS, so established pharma companies, but also medtech and SMEs, and also multiple biotech companies that have expressed an interest to join the consortium. And what we offer to the consortium is obviously the specific expertise in some of the platforms proposed, but also very strong regulatory expertise that we hope to bring to the consortium. Next slide, please. What we would hope and are looking for from interested public partners is obviously expertise in gene therapy and the proposed platforms, so a messenger RNA, nanoparticles, lipid nanoparticles, and the AAV and potentially gene editing space. But beyond the specific academic or in-depth expertise on the areas mentioned, also looking for collaboration of regulatory authorities as mentioned, patients organizations, certainly also the European reference networks, because I have a good understanding on and potential access to real-world evidence that would help the development of the consortium and other interested partners. Next slide, please. Oh, sorry, that's so. Should any of you be interested to join the consortium or want to understand better the how the consortium, where we are with consortium, or would have an interest to join, please feel free to reach out to either myself or Michael Linden. We would be more than happy to kind of introduce you in more depth.
Thank you. Thank you very much, Dirk. Thank you very much, Michael, for your presentation. I would like now to give the floor to Alperen.
Thank you very much. Hello, everybody. I'm Alperen Acemoğlu, and I'm a co-founder of the GlowCode startup company, and we are developing neuroprotective glaucoma treatment devices. Next slide, please. So, as you might already know, glaucoma is a disease where the intraocular pressure goes abnormally high and makes pressure on the optical nerve, and it is one of the most common causes for the irreversible blindness. The main problem here is even if patients use the today's gold standard treatment methods, still 15% of the patients go blind. That actually shows us glaucoma has another aspect, which is neurodegeneration. Glaucoma is not only about the eye, but also about the brain, and it creates neuroinflammation in the brain. In order to prevent this neurodegenerative side, we are proposing a non-invasive neuroprotective treatment method, which provides gamma-rhythm stimulation to the patients. And we have started our clinical trials in Turkey in 2 university hospitals. We have exciting clinical data. Next slide, please. So we are looking visual work potentials, web results of patients. When the patients, they come to the clinic, we took the results. Double click on the slide, please. And after using for 1 month Glocott wearable devices and getting this novel treatment method, patients improved their electrophysiological activity on the entire path from retina to visual cortex. And we saw these results almost in 30 patients right now. Next slide, please. And as a Glocott company, we are developing novel wearable devices for the treatment of the glaucoma disease. And our objective is to decrease the total number of permanent blindness cases due to the glaucoma disease. And within this project proposal, we would like to perform more clinical trials with the key opinion leaders from all over Europe. And we will continue developing and improving our wearable devices, and we would like to perform usability trials, and we would like to see the efficacy of the treatment in a larger scale. Next slide, please. So we are a startup company from Turkey. We have expertise on medical device development, and we have manufacturing capabilities with a clean room facility and sterilization center. We can be a part of other consortium in case of need. And we are actively in commercial activity in ophthalmology market. At the moment, we also perform, we have clinical partners in university hospitals. Next slide, please. So the expertise that we are looking for are the clinical center hospitals to perform and extend our clinical trials even further. And we are looking for key opinion leaders in both ophthalmology side and neurology side as well. And especially we are looking for experts in glaucoma and neurodegenerative diseases side. Thank you very much for listening. If you are interested in, please get in touch with me. Thanks a lot.
Thank you very much, Alperen, for your presentation. I'd like now to call Andras to join us on our virtual stage.
Thank you. Hello, everybody. Next slide, please. Our title. Yes, so I'm András Dinyer from Hungary. I'm professor in biotechnology, and next slide, please. I would like to talk about some challenges in rare diseases. I think you are all aware of that, but I think it's very important to realize that most animal models are not adequate or not even created for human rare diseases. So in my opinion, in vitro methods are very important, and in that case, human rare disease patient relevant in vitro methods. That's appropriate for lead molecules, gene therapy, prior to large animal preclinical clinical studies. And what we are doing in Biotalentum is exactly this topic for quite a few years now. So that's a way to de-risk costly failures with new therapies, also costly failures due to inappropriate patient selection or inappropriate preclinical clinical results, which would result in rejection by the regulators. And I think in this way, we are very much fit for quite a few potential consortia in this call. And I will show how our patient-derived iPS-induced pluripotent stem cell-based in vitro platform can provide quite an important tool here. Next slide, please. So we would like to do this de novo or banked rare disease patient human iPS lines to generate relevant organotypic in vitro models for the advanced therapy applications. We can also characterize, analyze in depth the models, test the ATMP candidates on the models, and providing feedback for the development process in preclinical stage and de-risking the clinical translation. Next slide, please. So who we are? So I started Biotalentum 17 years ago, and we are working with human iPS lines since 2009. And actually, I coordinated scientifically 13 EU FP6, FP7, Horizon 2020, mid and large size projects. And we generated validated complex 2D, 3D models for neurodevelopmental, neurodegenerative diseases, including several rare diseases with CNS phenotype. If you are interested, we extensively publish that, so you can find it under my name. Also, recently we are generating microglia-like cells, which is very important to incorporate in 3D CNS models because the neuroinflammatory, neuroimmune components are very important. We have also several projects where we make 2D, 3D cardiomyocytes models, and I'm also coordinating an ongoing ATMP preclinical study in beta cell Langerhans islets. It's not really the main topic I would like to offer here, but if somebody is interested, this is also within our expertise. So what kind of readouts to provide? We have high content imaging, functional calcium signaling, multiomics, electron microscopy, just to name a few, but we are very flexible to customize the process, because in a case of rare disease, I think flexibility and some innovativeness is very important. Next slide, please. So what we are looking for a consortium which would be interested to incorporate the in vitro models and assays into the pipeline. And indeed, if needed, we are also offering project preparation support, because with so many projects I coordinated, I also created my own consulting branch and workforce. So it's I can tell you that we are providing it also at very professional level. So if you are interested, please find us on the web and email me. Next slide, please.
That's
I think that's it. Okay, thank you very much.
Thank you
for the opportunity.
Thank you very much for your presentation. And we have our final speaker, Lorenzo.
Thank you for the opportunity. My name is Lorenzo Moroni. I am a professor in biofabrication for regenerative medicine at the Merlin Institute for Technology-Inspired Regenerative Medicine at Maastricht University in the Netherlands. Now, if you go to the next slide, please, I'd like to just give a glimpse on one of the activities that we can provide to a potential consortium from the work we do at the Institute, which is in the context of biofabrication technologies. These technologies have already been useful to create advanced engineered tissues that are nowadays being used actually to study other kind of therapies as a kind of advanced 3D in vitro models. And you see here some of the engineered tissues that we have been able to develop in the past decade, which indeed today we are also using to test, for example, other kind of drugs or medicinal therapies as a surrogate of the same tissue or organ in the patient body. These kind of 3D in vitro models could also be used to engineer actually the deliverable or the delivery of multiple biological factors in a controlled and targeted manner in 3 dimensions. If you go to the next slide, please. So the Institute is a technology-centered regenerative medicine institute, which means that our core business is to develop a number of different technologies that can be useful and serve the overall arching purpose of regenerative medicine, and in particular in terms of regulatory opportunities of ATMPs. And therefore, our daily business is to develop and characterize biological constructs able to deliver different biological factors via controlling multi-material composition, architecture, and disposition of the final implants we create. Next slide, please. In the specific context of biofabrication technologies and biomaterials, we have different expertise from both top-down approaches, such as additive manufacturing or bioprinting, as well as electrospinning, and from bottom-up technologies, such as micro nanomaterials that can be combined together with cells to create smart injectable solutions. Next. These are some of the main activities that the biofabrication technologies are centered around in terms of designing constructs that are able to steer cell activity, in particular stem cell activity. We continue to develop biofabrication technologies not only for the purpose of regenerating tissues, but also to understand degenerative phenomena. And in this effort, we have a particular interest in integrating neuro and vascular cues, as well as in understanding the immune response of biomaterials and the scaffolds that we create, not just to understand the fundamental science of it, but also really to engineer the immune response so that the final implantable devices can be more actively accepted by the host body. Next. When it comes to bioprinting efforts, you see here already a number of different activities that are either currently running or have been developed in the past, from organ-like models such as pancreatic tissues, cardiac or kidney in vitro models, or again thyroid models for 3D screening of chemicals that are present in our environment, to skeletal tissues and neurovascular models. We are also active on the development of new hydrogels that can better mimic the extracellular matrix of the tissues, composing the tissues in our body. Next. We are also active in the creation of nanoparticles that can be used as with multiple functionality in terms of targeting specific locations in the body and delivering a specific payload on that targeted location. Next. Finally, this is the kind of other expertise that we think we could be complementary to, so we can provide different kind of technology platforms and would be very intrigued and happy to collaborate with other stakeholders that have expertise in the biology of biological factors to be delivered in ATMP specific applications in terms of preclinical and clinical expertise in terms of regulatory framework, health technology assessment, and GMP production. Thank you.
Thank you very much, Lorenzo. Thank you very much all for your presentations. As a kind reminder, so if you want to contact any of the pictures, the presenters, you can either go to the pictures tab on the top menu, or just go to the agenda and you'll be able also to find their information there. All the presentations and also the recording of this session will be available later on IHI website. Thank you very much for joining today's session. We'll hope to see you tomorrow at the other sessions. Thank you.